Okay, so today we'll be seeing a different topic again. Like in this course, the structure, how we are going is first we'll discuss about the biology and then the application part, how engineers have used some bioinspiration uh, or they've exploited biology itself for for some application purpose. Okay. Similar way, like we'll see some applications over here because we have already studied about the different organ systems like respiratory, renal, and cardiovascular, everything. And something we'll see where engineers can do some technology so that it can be used further. But before going to that, some of the basic things you all know is we are made up of multicellular. We are all like at least there are around 10 trillion cells on an average. And these cells, which are multicellular, they will differentiate. We have seen in the stem cell how the differentiation occurs into different tissues, and the tissues, though all the cells will have the same genome, the expression will be different in the tissues, and tissues together they will make one organ. So we all know the organs which are there over here. Some we have already dealt, and the organ system like renal system, circulatory system, which contains both the blood as well as heart for pumping, and at last, the cells, tissues, organs, and organ systems, proper division of labor and cooperation among them will make one organism. So, essentially, what is happening over here is, as an engineer, we are seeing the multicellular system to work as a system. The organs are different components, and you have one system, just like say a computer system which is made up of different components, or a mobile device which is made of different components, or a tab or anything. Same thing, you are viewing the biology as one system. Okay, so for example, in the first module or in the second module, we had seen how the cells work. Okay, now we have seen how the organism work due to the cooperation division of labor of different organs. Okay, they all cooperate to make one system, and this system, all the organs is like cog wheel. We all know like cog wheels are very important in engineering systems. Like for example, you take a simple watch or a clock, that is also made up of cog wheels. Okay, like that, all that is a big network, and each and every organ system is connected with this cogwheel, and everything work accordingly. And if there is some perturbation in some of the organs, then the whole system there will be perturbation, and slowly there will be disease outcomes which will be seen. Okay, so everything depends on from cells itself. So even one gene over there. So in a multicellular system, you can see like all from organism to organ system to organism to tissues to cells. When you come, cells will have all the four different biomolecules. In that, mainly the nucleic acids. When you take the DNA, if there is even a small error over there, a mutation, it will show up in the organism, like the sickle cell anemia example we saw. Okay, so everything to work properly, it is very essential. Say, for example, the proteins, misfolded protein, that also can give diseases like misfolded protein, like the Parkinson disease or the Alzheimer disease concerning the brain. Okay, because of misfolding of the protein, the whole organism itself will be uh, not balanced. Okay, it will not show the proper healthy individual will not be there. Okay, it will be a diseased individual. So it all depends on so much of complexities there, and everything together, just we are viewing it as system itself, as an engineering thing. Like for example, if you ask a doctor views everything medically, what is working or not working or anything. If you ask a scientist, we ask how and why something is working like this. And if you see an ecological scientist or an evolution scientist, he will view the system as different things. But as an engineering people, we view it as a system. Okay, system made up of different components. Okay, it all works like this. So I told in the previous slide that it all works like a cogwheel, and all the organs, all the tissues and everything, the function is very, very important. And if there is some anomaly in that, causes a disease. Okay, it will not be a balanced, healthy individual. There is some disease. What do you call a disease? If some tissues or anything has an imbalance. That imbalance is called as pathology, and pathological condition reflects that is a disease. And in the world, when you see which are the highest number of diseases, this we have discussed. Topmost is concerning the heart, and then comes to respiratory. So one thing, the very important essential thing is both the respiratory, heart, or anything which is concerning with the disease in top level, in concerning the top 10 causes of death. When you see, it's all concerning the pipes. Pipe has been blocked. Like for example, the cholesterol, when it blocks the arteries, then heart indirectly, it will reflect the disease. And mucus clogs the respiratory pipe, then it will cause respiratory problems. Okay, so it all concerns pipes. So when an engineer or a plumber, for example, when you take the water, when it gets blocked, then you don't get proper water which will come at home. So what he'll do is he will essentially clean the pipe. Okay, that kind of mechanism is required. So that is the essential thing. But what we have to know over here is all the different organs, they are in division of labor with the in the whole system, and thus the human system or the multicellular system is able to work. Okay, for example, now if there is an anomaly taking place, because every organ is connected. Okay, I'll just have some water. So when some anomaly happens in some tissue or the organ, you provide therapy for that with the drugs, so that this recovers and the whole system works properly. Okay, this is what if you get a disease or anyone gets a disease, we go to a doctor and doctor gives some drugs, chemical drugs, which is helpful to influence the recovery of that particular disease of the symptom. Okay, now how how this drug discovery take place? Okay, how this drug discovery How, for example, now there are anti diabetic drugs, there are for obesity, they will give drugs. For hypertension, they will give drugs. For everything, disease, like for even an infection, they will give drug. How this drug is discovered, when you see, it's a very tedious process. Okay, so how it will take place is, for example, as a researcher, I will select where exactly that is happening. For example, uh, might be it is called, like, let's take the example of sickle cell anemia. There is a gene where mutation has happened, or for some other, say, a fever you have, and you want to reduce the fever or reduce the inflammation. This gene expresses this particular metabolite, or we say cytokine, for example, the chemical signals which will signal with the inflammation has to occur, with the pain will be there. Inflammation will lead to pain, so you will reduce the inflammation, then pain also will not be there. So, like this, the gene which at last, from what is the disease, when you know what is the cause, you will come to the level of the gene, and the gene, what protein it expresses, and can we inhibit this protein, then everything will be okay. Okay. So, most of the drugs work like this, and once you identify what is this by research, what is the cause for this disease, the causal nature of this disease, then for that you will also find a drug, maybe from chemical libraries or from natural products, or synthetically you will design in chemistry lab a kind of drug which can match a lot of computational things also will take place for this. And this is called medicinal chemistry, field discipline. And once you get plethora of drugs which could be possible, uh, it may reduce the disease, that you will do in the lab. Okay, it is called in vitro studies. Okay, and that drug you will test, say, for example, an infection of salmonella which causes typhoid. For this, I have discovered a drug, and that drug I will test whether it typhoid, it is actually antibacterial or anything. Okay, after this in vitro, after deciphering the cause for the disease, causation I'll identify, then I will have to club with the medicinal chemistry persons, and either from the natural, or he will synthesize a plethora of several drugs molecule, and even by computational methods we see what kind of interaction the protein is having or anything, and that will be tested whether it is effective computationally, theoretically, what we assume, whether it can be practically seen, experimental seen in the lab. In that, say, for example, 100, just arbitrarily 100 different drugs were synthesized at this level. At this level, less than 10%, maybe 10 drugs might be effective. Then I will see, because I cannot use the humans as a this thing. I will use animals, especially the mice or the rat, and there will be infectious models, animal models for different infections, like for Alzheimer's, Parkinson disease. They have monkeys too over there, and for some things, rabbits and mice studies can be done, and the drug will be tested on them. This is called as preclinical studies. Then it will go to, after it is found effective. Like, for example, as I told you, Here, maybe around 10% might be effective. So 10 drugs you have in hand, you test the 10 on the mice, but actually only one or two can come up over here again. From that, again, let's say 10%. So only one will come over here. One drug might be effective. Nine others may not turn effective. Okay, because the complexity of the multicellular system is very complex, okay, very high. Then this drug may then come to clinical trial. There are different phases of clinical trial: phase 1, phase 2, phase 3. By now, you might have been aware of these things, clinical trial, because the COVID vaccine also went into clinical trials like this. Only then the WHO gave the approval for them that it can be used on the public. Okay. After this, there's the same thing even for the vaccine. Okay. Drug or vaccine, everything has to go through this. They have to go through in vitro, in vivo, and then clinical trials. And clinical trial also will go in different phases. First, there will be less number of people, and then there will be a little more number of people, and then it may go to little more, and then it may come to market exactly after phase 3 trials are done. Okay, and how much participants are required for the clinical trial? When you see this, the preclinical, the animal study, phase 1, there could be 20 to 80 participants. From here, whatever was effective. So just see over here, so much of it's a very tedious process. Then another tenfold more people will be there in the phase 3 trial. Only if it shows in all these three phases, some will in phase 1 only it will go, some drugs will go phase 2, many will go phase 3, many will go. And if a drug is very effective, it qualifies phase 3, only then it will come to a review. For example, FDA is the Food and Drug Administration, not of our country, but US Food and Drug Administration, but it is having one of the stringent regulation. So many other countries also look for FDA, but we do have our own regulations or anything. Then it comes to phase 4, for example, the vaccine. Now, what all we are getting, Covishield or Covaxin and all, it is given as an emergency approval for giving it to people. So it's a emergency approval. It is not going through the proper phases where 1,000 to 3,000 or 1,000 plus participants over here in phase 3 or phase 4 you do, but it is given emergency approval because it's very important, because it might have qualified phase 1 and phase 2 trials. Okay, and how much time these things will take place? You see, say a drug discovery itself in the stage, okay, till the medicinal this thing, it will take around 3 to 5 years. This is a very small period which I'm telling. There are also, for example, 5,000 to 10,000 different compounds might be used. I give a very small 100 compounds for you guys, but over here, 5,000 to 10,000, you can see only 250 compounds on an average qualified. So preclinical, in vitro, and in vivo test will be done, and that may take 1 to 2 years. There is a short period, even 3 to 5 years and 1 to 2 years is when properly focused, these things will happen. Then, when it comes to this stage, the clinical trial, phase 1, phase 2, phase 3, and all, it will take around 6 to 7 years to complete. So, at last, 1 to 2 years for the new drug to come into market, and then promotion, all these things will take place, and the drug will be given by the doctor. So, if a doctor is giving a pill, it has gone into 5, 7, and 14, 15 plus years. Okay, so whatever we take from medical stores, a drug, it must have gone into 15 years of work. Okay, so many different scientists, doctors, clinicians, all are involved, and patients are involved, and so many stakeholders, other things, other management people, everyone might be involved to bring this drug to the market. So just say from 10,000, only one new drug can come. So it will filter out, filter out, filter out, and it's a very stringent kind of system which is there. Okay. However, can we make it less by our bio-inspired tools or anything so that we get more kind of compounds? Let's see that. Now, one of the example, this recent example, because we are aware of this, see the coronavirus vaccine. You can see the vaccine when it was discovered. You can see the typhoid vaccine has taken from 1880 to nearly 1990 or something. Okay, so nearly 100 years it has taken, more than 100 years. Okay, one vaccine to do all these things. Okay, and to come into market. Still, for typhoid fever, vaccine is still not very effective sometimes, but it all depends on different processes. I'm not going to it. So, see, the meningitis also is nearly 100 years it has taken, and polio vaccine has taken how much? 19, say 1910 to uh, 1950, 1910 to 1950. So 40 years might be here. So different amount of years has been taken. So it is not a very easy task, vaccine discovery and all. But see, because we accelerated and urgently we required coronavirus vaccine is only 1 year. Like for example, we had the first lockdown, and by the next lockdown in 20 to 21, we had the vaccine, and we all, most of us got the vaccine also already. 2 doses we might have got. Okay, so it is not only discovery, it has come to market. Companies have produced so huge amounts of vaccine, and this vaccine has been also given by clinicians. It's a humongous work which has taken place only in 1 year. It is great development in human history itself, you can say. Okay, so, how do we do this in biology? This is my lab. This is my PhD student. So you can see this is a biosafety cabinet. Okay, where she's. You can see both her hands are inside the cabinet, whereas her face is closed by this particular closing door, which is transparent. Okay, why I came here, I'm showing this is because there's called as a biosafety cabinet. When you all come to campus, you can come to my lab and visualize how this works actually. Okay. So what this also what happens over here is there is a filter, laminar filter, HEPA filter. Okay, it is a high efficiency filter, particulate filter, HEPA we call. And when the air passes, the outside air will be taken, and HEPA filters will filter it, and the whatever the air is there inside this machine, that is the cabinet, you have only filtered, no microbial load, and that kind of machine will be there. And you can see holes over here in this machine, so the air will from here will just flow into it and keep on circulating. So from outside air, the inside air, so from outside when the air goes inside, this will block. Okay, there will be a pressure and it will be blocked, and only the air inside will be filtered. Okay, so when you work with some organisms or some biological things like this, like in vitro studies, which was shown earlier, so you will have to work it with proper gloves and mask, and uh, very properly you will have to work, because these are biological things which can spread. Okay, it can be more toxic, or it can cause some disease or anything, or it can be when you do you take some tissue, say for example the kidney tissue of someone is taken while working, no contamination of any microbe also should take place, then you will not be able to work. So you use these kind of biosafety cabinets. There are different biosafety levels for different organisms, which I'll tell you later on in the module 6. But there's how in vitro works goes on. Okay, in the clinical phases, we saw in vitro works, and then if this works properly, then we'll go to in vivo condition, that is the animal studies. Okay, and now, because so much of technology has been sophisticated, like I even showed you, the coronavirus vaccine can be was done in only one short year, including the production of it. Now, with all the technology, all the engineers also will contribute computationally also whatever is required for any complex matter which our brain cannot comprehend, the computer can comprehend such kind of tasks. Can we now do personalized medicine? For example, earlier what we saw was one drug, and that is given for everyone. Like, for example, if you use paracetamol for when you get fever or any pain or inflammation or anything, at that time, paracetamol is used by all. Okay, say a 400 mg paracetamol is used by everyone. Whomever you know in the world, they all use paracetamol. But it is not effective in the same way to each and every individual who take paracetamol. That is one of the example which I gave. But like this, there are many drugs which may not be effective in the same fashion to every individual, because the complexity of how the individual is built up may be different. For example, we don't look same, but we all look similar. That's all. Two hands, two legs, one face, two eyes, one nose, one mouth, two ears, and like this. We are looking similar, but not same. There is heterogeneity in us. There is variation in us. Okay. So now, can we come ahead to make effective drug? Can we also shorten the drug discovery process, as well as give individually different drug which is effective to that particular individual? Okay, so instead of giving one drug to everyone, you can make different drugs suitable for that particular individual. Can we make this? How the technologically we have to go forward. Okay, there is a lot of research going on in this particular paradigm. Okay, one of the example, like historically, when you see traditionally the Ayurveda, which is there, is similar to personalized medicine. Not fully personalized, but at least it goes to a higher extent. For example, they see the physiology of the individual, how he is, like they call Vata, Pitta, Kapha, and they have a lot of classification. And in that classification, where this individual belongs, what could be the disease, and then they treat their disease accordingly to the makeup of the individual. So it is a kind of personalized medicine. So doctor has to diagnose the particular individual. Whereas the allopathy, now currently what we are having, can be diagnosed even when you tell, you make a very, say, arbitrarily, artificially using artificial intelligence, you make one app or some website. You give what all your symptoms. It can give you what exactly is happening. Okay, it is not so very this thing difficult process, challenging process, because the drug discovery process is very streamlined to that particular symptom. But now, can we go ahead with the technologies what we are having, and in future we may also get a more kind of technology where all the kind of engineering disciplines might be involved, where we offer each individual right drug which is for that individual's whatever disease is there. Okay, so we have to find the right patient and for the right disease, right time, and with the right dosage. Okay, for that, what is essential is you'll have to see the genetic makeup of the individual. In this, I'm showing you only genetic, but also the microbial things in our body. So, what is the genome of the microbes? What is the genome of that individual? What are the metabolic profile of that individual? How it should be ideally, and we can tailor each drug for each individual. Can we go in that particular direction? That is the motivation to go in that direction. What all we require here, I'll tell you simply. So before that, can we make, instead of the testing it on some animals or anything, can we make one chip where we grow the organs on a chip, small chip itself can grow the organs of that individual itself, not of some other individual, of that individual itself. So, see, for example, you can test drugs on the patient's cells only, same cells of the chip, if you can give, and it will also give the accurate data of the and even the dosing for the patient. Okay. So how can you do this? Personalized medicine, there are so many benefits. For example, you are reducing the time for drug discovery process. You're instead of clinical trial for every individual, so you're doing only for that particular individual. Is it suitable for that individual, him or her, to him or her only, whether it is suitable, and you're also taking the cells of the same individual and testing it. So it is very accurate for that individuals. You are not taking some animal or some other cells. Okay, you are taking the same individual cells, organs, and you can test. So exciting, is it? So how can you do this? So, what can other engineers, like of different disciplines, engineers of different discipline, like you might be in IT or you might be a mechanical or aerospace or anything, what can you offer? Okay, so sorry. So, what can be done for this is you can place the organ on a chip. Okay, so it will be like this, a small chip on which you can grow the organs. You don't need to grow one more human which is who is same for that individual, whereas you grow different organs on a chip. Okay, how is this done? It looks like somewhat like this. Okay, and it's a small slide. You would have. In your school, look at different slides, isn't it? Microscope or anything. Something similar kind of slides will be there. And in the slide itself, you can grow the organs. That is called organ on a chip. What is this? When you see it is an artificial organ itself. And it's mimicking the organ function. Okay, how your body is functioning, how the organ is functioning in your body, the same component. As when you view it as a system, you're taking the same component and you're growing it on the chip, and it's functioning just like that. And you can also view it, so it can be translucent, and you can view that whether it is happening, and it's having a lot of potential to transform, turn over the whole discovery process, which we saw conventionally how it occurs. Okay, so you don't need to kill some animals or anything. No ethical committee which is required, because it's the own cells in vitro you're doing, and with the same cells of the patient you're doing, and in a compressed kind of time you're doing this particular thing. So it's a great task. Okay, how you can do this? By microfluidics. So just like the circulatory system, here you have the fluid which is moving around in very low amounts. Okay, so it is essentially you're doing first if assume this dots over here to be the organs. All this what you're seeing patterns are plumbing. Okay, so you do a miniaturized plumbing essentially as an engineer, and you make the flow of in micro levels in very low amounts. Okay, why low amounts is more beneficial is because when you see the body, there is some spatial structure in the spatial, in the sense in the space there is structure properly, it won't intermix. Like for example, let's take the heart. When we discuss the heart in the cardiovascular discussion, we saw how the atrium and the ventricle blood will not mix, or as well as the left and right chambers also will not mix. And if they mix, it's a disease. Okay? Similarly, the fluids which flow over here should not get mixed. In microfluidics, they will not mix. Okay, in a very low amounts. This is the principle which is there of fluids. Okay, in a very low micro volumes, they will not intermix. It can flow in any direction. It may also come together, but they will not mix. Okay, the blue, for example, over here, if it is there, it will be as blue. Red will be as a red when they even go next to each other. Okay, because that is the property of microfluidics. Okay, that you are exploiting over here. And what kind of material you're making for the chip is mostly the silicon is used, and PDMS they call. It's a very cheap device. Otherwise, you can make it on glass, ceramic, polyurethane, polycarbonate like this. Even on paper also you can make. Okay, you can take a small paper, but everything should be sterile. Okay, sterile in the sense no other organism or anything, and that biosafety chamber you will have to work. But these kind of chips can be done. More popular among this is the PDMS, which is also a kind of silicon material. It's very cheap and uh, robust, so people use that, and they use other kind of materials also. And now we know a fluid in a low volume we use, and we make the channels plumbing, and we for the organs. Can anyone suggest what to do for the generation of the cells? or the organs. Now we know we do the microfluidics in these materials, but you need organs like over here. Different colors are given over here. P1, P2, P3, P4. There are organs. How do you grow the organs for that? Organ in the sense the cells essentially. How we can grow? Any idea? Anyone? Who will tell? You can guess. Anyone?
Sir, by giving an appropriate enzymes.
Not enzymes. Your cells you will have to grow them. How can you grow the cells? Like go to my previous classes and just think about it. What can be done? It is not very tough. If you had attended my previous classes, it's not very tough. So, say for example, even you can get the stem cells. You had seen in the previous class. Okay, and you can grow the cells over here. Maybe your heart, like a fish, it is not growing as a three-dimensional heart, but heart tissues are growing over there in a miniaturized way. You're making your lung cells, your tissue, heart cells, your kidney cells to grow. Cells are growing over there, okay? Or you may also take any somatic cell or anything and grow over there, okay? Cells are essentially growing, and you can make like you can take the cells and all the cells can grow in one chip itself or different chips if you want to test individually for the system itself. The whole cells can be grown in the chip itself, okay? And once you grow, you know the material in which it is there, what are the channels in which it can flow. And then if circulatory system you can mimic and you can put the drug and you can test toxicity of that if possible. Okay. This will be the organ on a chip typical thing where there will be different circuits like this. That circuits are the channels. You have done the plumbing and all the detection will be here, and what has to flow just like circulatory system will be given by the different small tubes, and they will flow over here. What all will flow, and your computers for that all the kind of sensors and everything is required, and you sense them, and accordingly display will be there, okay? And from this, what all you can do, you can reduce the animal testing, you can predict more effectively, you can reduce the time and just accelerate the discovery process of drugs. And individually you can do, and the applications are disease, you can model how this disease might have caused or even toxicity you can see, and multiple organs on a chip also can be studied together what is holistically what all is happening, okay? This one thing. Then now one more very important thing, okay? I think time is up. I'll stop here. So for the next thing I'll say tomorrow, okay? We'll see tomorrow. For this any questions, I'll stop here. If anyone have any questions, I'll be happy to answer you.